up to a week. This had been done for many years, and the discipline with doing it has disappeared. So by fault, by staff, residents, etc., 30% of our patients did as a fact not receive any prophylactic antibiotics. No intra-abdominal abscesses were seen in this group. All the abscesses were in the group with vigorous prophylactic antibiotics. And this leads to one suggestion to you, and that is that the prophylactic antibiotic, which is meant to prevent infections from the skin, eventually promotes infections from the large intestine. And I'm going to explain that to you. That's number one. The other thing is, prophylactic antibiotic treatment in surgery means high concentration in the tissues at the time of surgery. And I honor that. That is very important and probably also well functioning also in liver surgery. But as a surgeons, we have a tendency to think if 1 day is good, 2 days are better. If 2 days are good, 3 days are better. It might not well be so. The reason is that, and I will come back to explain that to you, when antibiotics are given during surgery, there is no salivation and no bile secretion, and no antibiotics will leave the circulation and the body and go into the digestive tract and kill the beneficial flora. And this is extremely important, and I will come back and speak more about that. A big step forward was done in 1985 when a surgeon and intensivist in Buffalo, United States, showed that cases who did worse were patients who had the combination of prophylactic antibiotic and parenteral nutrition. They did especially bad compared to others. Today we know a little bit more, so I'm going to reformulate that and say those who did badly or do badly are those who get prophylactic antibiotic for longer periods of time, and those who lack enteral nutrition. There is nothing magic with the parenteral. It's the lack of enteral which is important, and I will explain that further. What happens is that the antibiotics are secreted by saliva and bile. It goes down into the large intestine and kills the beneficial anaerobic flora, which controls the potentially pathogenic microorganisms and hereby allow these to colonize. The absence of a food stream further promotes the colonization. When the intestinal portal barrier is blocked, a stream of bacteria and toxins will hit the liver. And when liver is blocked, you know, each macrophage can swallow about 100 bacteria. At the best, 150. Each macrophage can do it, each Kupffer cell. But when they are full, they go to sleep, and there is no barrier, no protection anymore. So then the bacteria continue to the next organ. After the liver, it is the lung, and when the lung is blocked, it is, you know, the kidneys and the spleen, etc. We know today, it is recognized, that gastrointestinal tract is a source of bacterial infection, which is especially often seen in stress, in trauma, and in immunocompromised patients. The process by which bacteria leave the intestine and infiltrate into the body is called translocation. A lot of studies is done to the mechanisms of translocation. I'm not going to discuss that. I only want you to be familiar with the word. Under certain conditions, bacteria will no longer be whole inside the intestine. They will leave the intestine and infiltrate the body. And the mechanisms behind that are extremely interesting. However, mucosal atrophy, lack of energy, insufficient membranes, insufficient mucus layers,